Hi, I'm Talia Shaler. I'm a PhD student from the lab of Professor Asaf Vardi from the Department of Plant and Environmental Sciences. Now we'll talk about phytoplankton resistance to viral infection, or what happens when a virus meets an algae. So the sea is everything. It is the living infinite, quoted from Jules Verne's 20,000 Leagues Under the Sea. And indeed, the oceans are the foundation for our existence on Earth. They're a source for oxygen, they absorb CO2 and store it, and they also influence cloud formation and the weather. But the heart, the engine of the oceans are actually the tiniest organisms, like phytoplankton, bacteria, and viruses, the ones that Nofal just mentioned. So phytoplankton are unicellular organisms that perform photosynthesis. They convert light energy into chemical energy and oxygen. And they're actually responsible for 50% of the global photosynthesis. So 50%, okay, that makes them equal to all land plants and the rainforests. And in the right environmental conditions, they're able to rapidly divide in the water and cover huge areas of hundreds of kilometers in these massive events that are called algal blooms that can be seen even from space. Okay, this is a satellite image that captured the bloom. And I just want you to notice the sizes that we're talking about, the differences in sizes. So the cells are in the size of microns, okay, but the blooms that they generate is in the scale of hundreds of kilometers. And additionally, they're also the basis for marine food webs. Okay, they are like the grass of the ocean, and all other organisms are based on them. So they have great ecological importance, and in our lab, we study the same model organism, like Nofal, which is Amelia huxley. Okay, it's a unicellular organism that is a bloom-forming, generate these blooms, but actually their blooms are vanished within a few days once we see them. And the reason is that the cells are infected by a virus, this guy, that infects the cells and kills them. And yet these blooms reoccur year after year, suggesting that some cells survive the infection and somehow protect themselves from the viruses. And we wanted to know, how do they do that? What makes them different from the sensitive cells? And specifically, what genes do they express in a different way? So that was the question of my project, of what resistance mechanisms do Amelia huxley cells have. And in recent years, there were many discoveries in the tree of life regarding immunity and defense mechanisms that were discovered in like bacteria and plants and animals and humans. But this branch, sorry, the laser is not, this branch of unicellular eukaryotes, where my algae is there and the rest of the phytoplankton, the protists, remained underexplored. So after we will discover resistance mechanisms, we wanted also to know which genes are the ones responsible for the resistance and define maybe a set of markers genes to detect these cells in a mixed population. Later on, take it back to the environment, back to the ocean, and using these marker genes to detect this rare state of cells in a natural infection. And just in brief about the challenges about working with this kind of organisms is that they're not the typical lab organism for biologists. Okay? They're not like mouse or E. coli, where we have already great tools to investigate them, like genetic modifications and resolve genomes. Okay? So this is part of the challenges that we are facing. But what I did was to take Amelia huxley cells and infect them with a virus in the lab. And we observed typical infection dynamics. What you see here are cell counts and viral counts. So in the green line, you see a decline in the number of the cells that is followed with increase in the blue of viral particles, okay? While in gray, the uninfected culture remains stable. But then we waited for a bit longer, and we saw that their cell cultures were able to recover from the infection while there's still high abundance of viruses in the same flask. This suggests that the cells gained resistance to the viral infection. We further inspected them and infected the recovered culture again with the virus, and saw here in the green line that the recovered culture with the virus grew in the same manner like recovered culture without the virus, meaning that they are indeed resistant to the viral infection. And throughout this time series of 50 days, I also sampled for RNA sequencing. That means mapping the gene expression of the cells during this transition to resistance and trying to detect which genes are now expressed in the resistant cells compared to the original sensitive cells. And what we discovered is about a bunch of 2,000 genes that are significantly differentially expressed during the time. And we grouped them according to their change in expression through time. So what you see here, this is a heat map of gene expression through time. Each row is a gene, and it shows the relative expression of it in the different time points. So here, this group is T0 to T3, day 0 to day 3 of uninfected cells, the same time points, 0 to 3 of infected. And these are the later time points of day 19 to 50, where the cells are resistant. And what we discovered are these groups of genes that are unique to each phase. For example, in this group, you see there is a group of genes that is upregulated only at one time point. This is infected cells at time 0, so 30 minutes after infection. And it represents a very immediate response of the cells to the virus. And indeed, we also saw a lot of genes that are related to signaling pathways. We have these two groups of genes of either upregulation or downregulation of genes through infection. And then we have many genes related to protein translation, photosynthesis, energy production. That means that the virus is really remodeling the entire metabolic pathways of the cells in order to turn the cell into a viral factory. And finally, we detected this group of genes that are upregulated only in resistant cells, see, only at these time points, suggesting that these genes are linked to the resistance. And we wanted to further validate it. So what I did is a meta-analysis with 5 other isolates of resistant Amelia Huxley. So what we have here is basically 5 different data sets of other resistant isolates that I checked which genes are upregulated there. And I wanted to see if there is a shared, mutually shared group of genes that are expressed in all resistant cells. And what I saw is that if we focus on this group, is that each line here is a different resistant isolate, isolated again from different places in the ocean, processed by different labs. When I'm looking at the genes that are expressed in these resistant cells, you're looking here to the right, most of them are clustered in the 4th group, in the one that is related to resistance. And if we're also looking at specifically the 200 genes that in the dark color, these is a group of genes that is expressed in all the resistant isolates that I tested, meaning they are a really strong core group of resistant genes that we can apply as markers to detect these cells later on. And to what these genes are, so we detected many genes that are related to cell wall glycan modifications and sphingolipid remodeling. That means that the cells are expressing genes that can change their sugar and lipid moieties on the cell membranes. And this is like the first step of recognition between a cell and a virus. We also detected some genes related to nutrient uptake and metabolism of nitrogen, phosphate, and CO2. And again, we know that resistant cells, they have a burden of being resistant. This is probably the reason why they're in so low numbers in the original population. So when we see that they're upregulating genes related to metabolism of core nutrients, that might be the gene expression profile of them being resistant, their cost of being resistant. And finally, we also detected some pathways that are related to innate immunity mechanisms that are conserved across the tree of life. So we know them from human, plants, and bacteria. And now, for the first time, we detect them also in phytoplankton and in conditions that are related to resistance and pathogen recognition. So for the next, I'm planning to use these gene markers that I identified in a different method that is much more sensitive. And it's single-cell RNA sequencing. So again, now instead of mapping the gene expression profile of the population, I'm going to see the gene expression of individual cells and maybe able to track and detect these small population of resistant cells in a mixed population like the one that we have in the ocean of the natural blooms. And we actually already have the data and also are analyzing it. from bloom from Patagonia that were collected 2 years ago. So with that, I would like to thank you for your attention, and my lab and PI, and also the organizing committee for giving me the opportunity. So thank you.